All right, so I'm from Swift. I'm going to try and make this a lively discussion. I have lots of next-gen sequencing stories. I grew up on 454, and then I switched over, and I did solid. And then I did ion torrent. I've had both R&D roles and commercial roles. So I really know how to pick the winning next-gen sequencing platforms. So I decided actually what we'll do is do next-gen sequencing libraries for all the platforms. And that way I don't have to worry about whether I picked the right one or the wrong one. So we're going to talk about libraries today. I have lots of examples. I may go through some of the slides towards the end very quickly. If there's something you're seeing there and you want me to back up, I will. But in the sake of time and as we're getting late in the evening, I don't want to put people to sleep. So we're based in Ann Arbor. We have nothing to do with the University of Michigan. But what I can tell you is when they lose, the town is depressed for a day or two. Okay, so we have 4 different products. I actually want to thank Matt for giving me an intro for the Methylation, because we'll talk about that in some more detail. But these are the products, the 2S. So Excel NGS is our brand name. My marketing guy always says, you got to say that. So I said it, so I'm good there. I'm going to call it 2S. What 2S means is double-stranded DNA. Everything double-stranded DNA, that's what we do. I'm going to show you how we do things differently. And what I really hope is to get you guys thinking about library preps. And I was going to make a slide, the proverbial garbage in, garbage out. But that's what library preps are about. And if you want to talk about sequencing costs, you can eat a lot of money in your library preps if you're not paying attention. The other, when we're talking, say, about methylation, in some of those preps, you need a microgram of DNA. So you need a ton of DNA. So library preps are actually kind of key to the whole part of next-gen sequencing platforms. Do it well, you're going to get really good data. Don't do it well, you're going to spend a lot of money on data that you're going to throw away. And if you do it poorly enough on clinical samples, and you consume those clinical samples, you never get it back. So that's my little preaching there. I'm going to talk about amplicons and what those are about. We'll talk more about methylation, because you can never have enough of that. And then there's one technology called 1S. Here, what I'm going to show you that it's okay to boil your DNA in sodium hydroxide. Yes. And I'm sure Vince has taught you to do that often. So I actually borrowed this slide from the Baylor Genome Center. This is from Donna, and I really like it because this is the trend of what we see in the industry. So this is the 10th year for the course. And what you see is that, way back when, we were using micrograms. So I had a front row ticket to the sequencing of James Watson. So when I was at 454, over beers, my son, over beers. When we sequenced Watson, we were first to sequence a named genome, named individual. And back and forth, we're trying to figure out how many micrograms we used. We used so much DNA of Watson on the 454 platform, we actually had to bleed him twice. We actually had to come back down and do a second blood draw because we used all the DNA in the first blood draw.
Did you guys nebulize the DNA then?
It's everywhere now. So with Watson, essentially, we used about 20 micrograms of input to sequence him. And that's a lot. Now what we can do is we can do a whole human genome at 30x with about 10 nanograms. So we've made a lot of improvements, and by we, I mean the community. All right, so a little education here. You may hear vendors talk about 100% conversion rates, and they are lying to you. They are not telling you the truth. Vince may not tell you the truth either, but that's something else. Conversion rates, when you talk about conversion rates, where you get hammered is actually in your your size selection. When you fragment the DNA, if you're not doing it enzymatically, if you're using a covirus, that's about the only way to mechanically shear it. It's like putting a little mini hand grenade in your Eppendorf tube. You've just shattered, blown apart the DNA. You will lose a lot of DNA there. You may not care if you're dealing with a microgram. You will care if you're doing single cell or 10 cells. Really problematic. Size selection, well, the Illumina platforms all need that nice 150, 300 base pair inserts. PacBio, they also have size selection too. They just want it to be kilobases. So you lose DNA there. Type of sample that you're using, you'll lose DNA there. And then, as Matt was pointing out, your depth of sequencing, if you're doing shallow coverage, you may not see that you're lacking complexity. But if you're going 20, 60, 100x coverage, you're going to notice you have those duplicate reads. So critical factors, I put this in here if we're talking about cancer. We will actually talk a little bit about cancer. You hear all these folks, oh, you can use 1 nanogram of input and we're going to get you down to 0.01% detection, right? Mutation frequency. It's super hard to think how that's about because you only have 3 chromosomal copies with that mutation on it. So unless it's a gene expansion or something like that, you have to really, you guys can pull the bullshit factor on people, right? When you hear vendors talking about getting ridiculously low cover or detection of mutations, keep the math in mind and just say that that's just not possible. So, did I say, I swore, didn't I? I'm sorry about that. Now this is why we do things differently. This should look very different than any library prep you've ever seen. So we start with double-stranded DNA in this one instance. And the first thing we do is we dephosphorylate everything. Right? So we're going to take the phosphates off both the 3' and the 5' end. This helps eliminate chimeras, and it improves our ligation efficiencies. Then we're going to repair the 3' end, and later on we'll repair the 5' end. And by repair, I mean if the base is damaged, we're fixing it. Then we're going to come in, and this is also very unique about us, is we come in with a P7 adapter. And then we come in with a P5 adapter. So we're doing sequential ligation of our adapters. And I'll show on the next slide why you may care about something like that. So in the end, the base composition is identical to Illumina library, but we've gotten there very differently. Because we get there differently, it means you can do different things with our technologies. So this is then, on the right side is sort of the traditional prep. This is what Vince is teaching you how to do, and we want to teach you how to do it this way on the left side. So if you look on the right side, essentially you're going to keep those phosphates. You're going to come in and do your A tailing, and then you come in with those Y adapters, forked adapters, a lot of IP around those things. And then you do your final library generation. Why this is really different is you're going to get a ton of adapter dimer. So one thing that you'll hear is, well, if you're doing low input, you have to titrate your adapters. That can be really problematic, and especially when you're dealing with very low inputs, a lot of times you can't even quantitate it. So you get these runaway reactions of adapter dimers, and that diagram is showing you why. We have very low amounts of adapter dimers. So we're doing things differently. Keep this in mind as you look at your own experiments and how you're setting things up. All right, so circulating free DNA. Hopefully everyone here has heard the hype about liquid biopsies. It's like the promise of cancer sequencing. It is quite powerful. It looks like it has a lot of promise, but it still has a lot of basic science to be done. So the concept is, instead of sequencing your tumor, we can take a blood draw from you, look at the circulating free DNA that we'll isolate in the plasma. From that, we'll do deep sequencing of it, and we're going to look for point mutations. Later on, I'll show you how we do methylation studies at that. Point mutation in P53, right, or whatever associated disease marker, you can then monitor how well someone's progressing with their treatment or not progressing. or maybe you see drug resistance. So it's a very powerful way for the physicians to actually monitor your treatment. In this case, we're using 10 nanograms of input, and we're making a PCR-free library. So that's kind of impressive. Usually, if you talk to Illumina or some Kappa, they're going to be up around 500 nanograms to do a PCR-free library. This is what the data looks like. So we have good coverage across the whole human genome. And then, again, this circulating free DNA, it's wrapped around the nucleosome. There's one wrap around the nucleosome. It's 169 bases plus or minus 1 or 2 bases. So because it's enzymatically digested, we don't have all that damage from fragmentation, and it's the perfect insert size for Illumina sequencing, allowing us to have that 90% conversion efficiencies. So we're not at 100%, and 90% is about the best we do. Usually we are between about 50 to 90%. This is the best case scenario we have for conversion rates. And it's primarily due because the insert size are perfect. Okay, so a little science biology here. What we have is the 2S prep of just germline DNA over here, up top, and then with PCR at 10 nanograms. And these are Picard plots. Hopefully you've seen them. You're looking at your base composition as a function of GC windows going across. And what you want is that sort of nice smiley face. You don't ever want to see a frowny face. And I'm talking about these dotted lines here. See a frowny face, and that means that you've got a lot of GC bias. So as you can see, we add PCR, we're starting to get less of a grin. Now when we look over at the circulating free DNA, what you should notice is that the high AT parts of the genome are missing. And that's PCR free. So we're not losing it because of PCR or anything like that. We're losing it because it's just not there. And so it's just sort of a little side observation that when you do look at circulating free DNA, you're not getting the full genome back. All right. So X10 sequencing, HiSeq-X. This work's done out of Baylor, led by Donna Musing. And I'm going to show two different experiments. Primarily, I'm going to show these experiments to talk about more GC coverage, evenness, completeness of genomic coverage. And in this case, what we have is we have the Swift PCR-free at 200, 500, and 1,000 nanograms. And we're going to compare it to the TrueSeq Nano at 100 nanograms and 1 microgram. And you can see our duplication rates should be very low, but we are cropping up, and if you are still awake, you see at the 1 microgram we're at 18% duplication rates. How can this be? Ends up that if you read our specs, which Baylor did not read our manual, happens more often than you know, that we max out at 500 nanogram inputs. So they just overloaded our library prep, and we don't have enough adapters to actually utilize all the DNA present. So when we look at the GC coverage, we have our 5 data points here. You can see the TrueSeq is in the red, and they're dropping pretty precipitously there at the high GC contents. While for the Swift libraries, we're staying both pretty flat across the genome, and also we're covering more of those GC bends. You get one question, one.
Go, Jimmy, go.
Is it actually going up at 90% GC?
Yeah, and I think that's just an artifact, right,
I mean, my point is,
of mapping.
I think the ligation thing's good, but the other thing associated with this back in the day was the polymerase you were using as well. The DNA polymerases for the amplification. This is PCR.
This is PCR free.
Yeah, so wow.
Was there a question?
No, I just mentally spew stuff, so get over it.
And it's not over.
So then this is almost like a cluster formation issue, or no, it can't be that either. So what the hell's going on?
They're using the TrueSeq polymerase, and we never do.
But this is PCR free, right?
Not the right one, right? Because that nano is not.
Nano's not PCR free. That's
The-
right.
The green is.
Yeah, okay, so that's right. So when they says TrueSeq Nano, that's their
All right, we're moving on. I've got 110 more slides to go. Just kidding. Kidding. Maybe I'm not. This is actually just another way of looking at coverage. So Baylor, a lot of other places. Actually, Rick Lifton will tell you that sequencing the whole human genome is silly because we only understand the genic regions. You can argue with him or not. He's very passionate about it. But this is just, again, looking at the coverage, and we're covering the omen genes with high fidelity here. Now, the next experiment was actually for ovarian cancer. And I love this slide because they had a bunch of ovarian cancer trios that they wanted to sequence. They were out of material. It was sort of a kind of a retrospective to a retrospective study. And then 2 of the cases, they didn't even know how much DNA they had. They just washed the tubes and went for it. So low inputs, Vince has used it. You know, when you get below that 20 nanograms, you're left with, do I want to burn 10% of my DNA to figure out how much DNA I have, or do I just go for it? In this case, they just went for it. I'll highlight that the 1770 is actually one of the samples that was a tube wash. And for the most part, you're seeing very good sequencing generation, and for the most part, very low duplication reads. So, which is actually phenomenal. We're sequencing deeply, and we're for the most part below that 20% duplication reads. See that we get good coverage. As you look at coverage models, what you want to do is look at how things look at 20 and 40. Right? As you add depth, are you still maintaining good coverage? So don't ever get fooled by the 1x coverage. Everything looks great at 1x. Then when we look at GC, now remember this is where we're adding a lot of PCR. You can see that our curves are pretty much, for the most part, staying very, very similar to our PCR-free GC coverages.
Are you guys using the PCR here?
The polymerase? Secret. And it's not kappa. Not kappa. Kappa has a good enzyme. I'll go on record for saying that. But kappa's enzyme isn't necessarily the best across all applications.
And so NAB has a really good enzyme as well.
There are a lot of good enzymes out there.
But you guys are not doing enzymes.
What's that?
Are you guys doing enzymes as well? Or?
No, we're not evolving any enzymes. So, and for any future, we use pixie dust, okay? So, secrets. Again, we're looking at the gene coverage here, so just the genic parts of the genome, and you can see that the Swift is doing very well. 2 of these are actually the low wash, right? Or washing the tubes. So that makes sense as to why they have very low coverages, because they were just starting with very low amounts of input. So HYBE, anyone going to be doing HYBE here? Exome types of enrichment? Maybe, maybe not. Yes. Effective way to do a lot of samples. Has some issues with it. One of the big issues are need a lot of input. Still, you need a lot of input. Another big issue until Swift came along was FFPE samples were very challenging to utilize. So what we have here is our various inputs up here. So cell line FFPE, do library amplification, hybridize to the probes, capture, enrich, and then do a post-capture amplification if needed, and then sequence on the Illumina platform. We're comparing to kappa on this. This is cell line DNA, so nice, pristine DNA. And the big thing to point out is the duplication rates in this column. We're going from 100 nanograms down to 1 nanogram. And you can see that for the most part, kappa, I mean, when we get to 10 nanograms, kappa is ballooning on those duplications. Their coverage, though, is fairly good going out here. Well, kind of. They sort of actually start nosediving pretty badly. When you look at this, this is what I was talking about. You know, you get decent coverage at that 2x coverage, but they have no depth of coverage here. And so, again, as you look at your data, what you want to do is look across your coverage as you add sequencing depth. Make sure it doesn't fall off.
In each of these comparisons, the total number of reads you're getting per run is the same. You're equalizing everything else.
Yeah. So we're normalizing down to, because, right, then you get double the data and things go haywire. So that's a good point to. In this case, we're looking at FFPE. So FFPE, DNA is heavily damaged. And by heavily damaged, in some areas, they overfixate. They'll fixate for a weekend, and the DNA is pretty much useless. And so with that, all that DNA damage, again, if you think about making a library, and you're only fixing the 3' and you never fix the 5', how are you ever going to use an FFPE sample? So again, this is where we think we have an advantage because we're coming in and fixing both ends. And the data here, I think, talks to that. For kappa, we could never get them to work at the 1 nanogram or some of the 10 nanogram inputs. Again, you're looking at 10 nanograms down to 1 nanogram. Spectrum, these are clinical samples, different tumor samples. That's spectrum, and this is Horizon Diagnostic formalin fixed that you can commercially buy. So you can see the duplication rates, very low for us. It balloons for kappa. We're at about 20% with 1 nanogram. That's good. It's not great. But in a lot of cases, if you just have 1 nanogram input, that's what you're going to go for. And again, it's these coverage metrics that you really want to look at. You want to be staying up in the high, ideally in the high 90s, 80s acceptable. When you start getting below 80, that's when that variant calling is going to be challenging. If you're sequencing FFPE, you're most likely looking for a somatic mutation. So you're going to have to do a lot of deep sequencing to find that rare variant or mutation. Okay, I'm showing this because the next few slides are actually going to talk about Nimble-Gene enrichment. It's just, again, a series of controls, same trend as you've seen, 1 nanogram to 100 nanograms. You can see that kappa has the higher duplication rates, and that, again, you're looking for those coverages as you go higher. I think I've hammered this one home for you guys. Now, these next slides I think are pretty exciting. This is work that we've done with Silicon Biosystems. Silicon Biosystems is an Italian company. You don't hear of too many biotech companies out of Italy. And they have a really cool device. It actually uses this electronic chip. It actually, interestingly, it looks like an ion chip. It's small, it's an overgrown postage stamp. But this slide essentially is showing you what happens when we talk about cancer sequencing. I know Elaine's going to talk about this in a lot more detail, so I'm going to keep it very basic for myself and what I'm showing about. But when you do tumor sequencing, what you're doing is you're just taking a curl off your FFPE sample, and you're going to just sequence everything. What you're hoping for is you have good cellularity, so tumor content, ideally you'd like to be about 50%. A lot of times you find out it's 20%. So that means instead of sequencing something to 20 or 30x coverage, you're trying to sequence it to 100, 200, or in a lot of cases, if you're doing Amplicons, 10,000-fold coverage to see those mutations. So what Silicon Biosystems has done is they've created a way to take an FFPE sample and actually separate the stromal normal cells from the tumor cells. I'm not going to be able to show you that in any great detail. They have a nice website. They have a great video on it. Their instrument, actually, it kind of looks like a PacBio instrument. It's very large. But I would encourage you to go online if you're interested in this. Essentially, you're taking your FFPE sample, you're loading it onto this chip, and they're going to separate, based essentially on charge, antibodies into the stromal healthy. That's going to be blue through the next few slides, and the tumor will be red. When we come out, we're only going to have about 150 to 300 cells total. So we're talking a couple nanograms, and the caveat here is it's all FFPE. So even though we're separating the cells out, the DNA is still heavily damaged. So those genomic equivalents, those copies of the genome are actually lower than 150. Now I'm pretty excited by this data. So this is, there we go. This is our unsorted cells, 300 cells. What we did is we've used our 2S library, we've made a library, and we've done load genomic or whole genome sequencing low pass. So we're about 10x coverage, and you can see that you can't detect any, you can say all the chromosomes are there. That's about all you can say from that type of data. What we're really looking for is copy number. And the only way you can get copy number is if you can separate out the tumor from the normal. And in this case, this is what the tumor cells then look like. So this is low pass coverage. So one of the few experiments people know about is NIPT. NIPT, right, aneuploidy, you're looking for chromosome 18 duplication events. It's one of the first applications ever created on the solid platform because you just need a 35 base pair read, and you only needed 100 million reads or so, and you just would put them across the genome, and wherever you got additional more coverage, that just meant you had more copies of that chromosome. So you can use low pass coverage to actually look at copy number, but you need a pure cell population to do that. No way you can do that with a normal tumor cell or sample. So this is what it actually looks like. This is what the normal looks like. And again, this is what the tumor looks like when everything's separated out. So pretty cool. Elaine can tell you what it all means, though. Now what we did here is we took those samples, additional samples, and we're doing that Nimblegen medexome enrichment. And what we have is we have our tumor. Boy, none of my colors came through. We have the tumor up here, the normal here, and then unsorted. You can see the number of cells that we're starting with. The coverage of our exome is excellent. We're in that 95 and greater percentile. That's exactly what you want to see. Percent reads on target is also very good, and then uniformity is quite good. What's not so good is the duplication rate over here. So we've oversequenced these samples. Not surprising. All right, so note to self, don't sequence these so deeply, and you can still get the good coverage. So what we can do is, these are different preps, but they're technical replicates, so we can combine everything, and this then shows you that our mean coverage is 65 for the tumor, and normal is just above 20. So now we can do variant calls. Now remember, we're dealing with pure cells, so we don't have to look for mutations at the 5%, right? Those mutations are going to be either at the 50% level to 100%. They could be in between because of copy number gain issues.
What is percent uniformity?
Percent uniformity is, in this case, the percent uniformity I'm familiar with is 20% to the mean. So if you have 100 targets, how many of those targets are covered to the mean coverage of 1,000? So if I sequence something to 1,000-fold coverage, I want all my targets to be covered to a minimum of 200x coverage. 20% of the mean. This is normal. If you do look at your exome, this is why people say you can never do copy number with normal or with exomes, because you look at it and you're left with, there could be copy number here, maybe, I don't know. But when we look at our tumor cells, because cancer is definitely a very different beast, you can quickly see that we have copy number gain, or LOH here in the yellow. I apologize that some of my colors didn't come through. But look here, this gold kind of color, this is where we have copy number gain, right? So we're not right at 50%, we're more at 75% or 25%, indicating copy number gain. So you may not feel that this is a big deal, but this is kind of a big deal. This is a sort of a breakthrough in sequencing. Now the last slide for this study is we're looking at point mutations, right? So those P53, those really deleterious mutations. What we have is we have great concordance between ion torrent, our own Amplicon panel that I'll present in the next few slides, and then our exome data. And these are the various mutations going across. P53 here, I think I saw an EGFR is right here. And you can see that, for the most part, again, we're looking at a pure tumor. So the frequencies shouldn't be at 5%, 10%. They should be at something like 50 or 100%. Now what's interesting is there's a P53 mutation. I grabbed the wrong slide. But okay, in this case, what I was going to point out is that for ion torrent, they actually had a low frequency mutation. It wasn't a mutation, it was just a germline variant that looked like a mutation because it was coming in below 50%. Anyway, that blew that one. Okay, so I have 15 minutes and we're going to try and blast through a bunch of slides here. Amplicon sequencing. If you want to screen, do validation, Amplicon sequencing is your friend. As Matthew was saying earlier, this is a cost-effective way of doing a lot of samples on your instrument run. The thing that will bite you, though, is when you multiplex your amplicons, that means you have to order a lot of indices, right? Because you have to barcode all your samples individually. So if you're only sequencing things once, it could be a very costly way of going because you have a setup cost. Do thousands of samples and you're only looking at 100 loci, this is the way to go. So with us, it's a single tube. It's multiplex. We're currently about 600 amplicons in the pool, and we're pushing up to 6,000 amplicons in one pool. It's a 2-hour workflow, takes 10 nanograms.
What would be adjacent amplicons then, right?
What's that? You mean overlapping?
Or adjacent so that you would go around the products, because that's the thing with the amplification.
Hold that. We'll come back to that in a moment. Again, all single tubes. But as Vince was just asking, so P53, we have the entire coding region in one tube. So you have your exon, I'll show this in a couple slides, and multiple amplicons going across it. We figured out that usually when you tile amplicons like this in one tube, you get runaway reactions, right? You get these little, between my fingers, right here, the first digit, you get a runaway reaction. We figured out how to tame that so you don't get runaway reactions or super amplicons that can occur. These are all the various panels, and what I'm going to talk about is tell a great story out of the clinical lab on 56G and the sample ID panel. Our P53, again, it's the coding region of P53, it's 21 amplicons. Here's the size. It's smaller for FFPE samples. When you're dealing with FFPE, you rarely get DNA greater than 200 bases. It's about 150 bases plus or minus. If it's been overfixated, it'll even be shorter. Or 56G, that's for 56 related cancer genes. Number of amplicons is 263. The P53, all these amplicons are just a subset of what's found in 56G. So this is the data I was talking about. We have the gene down here. These are the various exons. And here's our coverage, these towers. And you can see where we essentially have, say, 2 amplicons here, and they're overlapping one another. You get that step-like function. And so we've been able to figure out how to sort of tame the PCR process here and be able to put multiple primer pairs in one tube. I was on the ion R&D team, so I look at this with mixed feelings, but we compared ourselves to some data generated by NCI for these 15 samples, and we came back and reported that they had 3 false positives in their P53 data. That's a huge false positive rate out of 15, right? And we took a lot of work on both sides to figure out that indeed it was a 9-torrent error. We could talk about sequencing errors all day.
How do you know that yours are not false negative?
Well, because when you go through and you look at the ion data, you can see that it's actually it's a sequencing artifact, right? So you see it in one direction, you're not seeing it in another direction. I know that was in one case, but that's a good question. None of the colors came through except for P53. This is 56G. The colors came through, you would see that there are about a dozen other genes where we have these overlapping or tiling amplicons going across it. Again, it's a fairly large panel. It hits a lot of the clinically actionable genes found in cancer. We use the Horizon diagnostic, so as you think about setting up your labs, always good to get good controls and run those controls frequently and often to make sure that your laboratory processes are working. When you're dealing with experimental samples, You think they all should work and they don't work. Go to your control. If it doesn't work, right, then you know you have a system issue. If your control is working, then you have a sample issue. A lot of times I was just talking to Elaine, I will share this one. The DNA came in without a buffer, right? They couldn't get anything to work. So these things happen. It's amazing what people, the types of samples you'll see.
Yeah, I was dumbfounded by that one too.
All right, so the horizon, why we like it, is it has these 10 mutations going from a 25% frequency down to 1%. When we do replicates, n equals 10, you can see that our standard deviation to the actual is very, very tight. The Excel Amplicon ID panel, again, if you're doing a lot of human genome sequencing, by a lot I mean hundreds of thousands of samples, you want to uniquely identify your samples, prevent sample swaps, and I'll show you why in the next slide. In this case, it's a nice publication out of Genome Medicine where we took 104 or 96 of their high minor allele frequency. So these are SNPs that have a high minor allele frequency across populations, and you look at the permutations of what alleles people have, and you can genetically fingerprint them. Then we have 8 specific SNP or sex IDs in there. And we can take this panel and we can spike it into 56G. And we can do this because we can have a spike in of the sample ID primers at 4% the frequency of the 56G primers. Right? So they're low frequency. So when we look at our cluster on the Illumina platform, the vast majority of reads will be 56G, but there will be a couple of the sample ID panels. Something to think about. So as we look for doing cancer sequencing, we're going to sequence a minimum of 5,000x. Often you'll see 10,000x. By 5,000x at 4%, we're going to get 200-fold coverage for these germline variants. And you only need about 20 to 50x coverage to confidently call a germline variant. So we can call our germline variants here and look for our somatic mutations in the data there, all from the same sequencing run, all from the same PCR reaction. And this was helpful because when we did a project with actually a lab not too far from here, we did 56 ovarian cancer tumor normal pairs. And just to give a snapshot of it, these are the number of mutations per tumor. So you can see that in 17 cases, we detected no mutation across our 56G. And a vast majority, or about a third of half the samples, we're just seeing one mutation present. What you tend to see is between 1 to 3 mutations present in lab samples. It's rare that you see someone with 8 mutations. I mean, that's a really sick individual. And then as we look at it, these are the various genes, number of mutations in time. So ovarian cancer, the predominant gene is P53. And our data supports that. Problem was we had 3 tumors that just were not giving us the data was befuddling. So when you do variant calling, mutation calling for tumor normal, you compare it to the normal, your tumor to the normal. It's sort of it's an internal control almost. And nothing was working out right. I mean, we're seeing way too many mutations or not enough. So we used our sample ID, the spike in data, to look at what was going on. And what we found essentially was that their tumor normal pairs were swapped. And these 3 cases, the color diagrams show you how they were swapped. What I'll also point out in the data I'm about to show is that we can also look for LOH. So loss of heterozygosity, a phenomenon that we do see in cancer. It's not frequently reported or really characterized. You don't see it in a lot of the papers, but I think we'll start to see it more and more frequently.
Can I ask you a question, just to understand how the sample IDs thing's working? So because it's not like you're taking an independent sample from these patients to look for these variants. What you're saying, I guess maybe I missed it, is the idea that since for every sample in this case you have tumor and normal, you should see concordance in the variants. So this will tell you that your pair between tumor and normal is correct. It doesn't tell you necessarily it's from the right page, when you can still have a pre-sample switch.
Exactly. So what we're looking at, these columns are all of our 100 SNPs. These are tumor normal. These are correctly paired. So white is wild type reference. Green, I always get this wrong. Blue is homozygote variant, and green is heterozygote. So you can see how everything lines up nicely here. These are the 3 pairs where things were not looking so good. I'm going to show this in another slide because it's way too much data. But you can quickly see that there's, loss of heterozygosity. Okay, maybe, but what's going on here, right? That this shouldn't be happening, right? So something clearly is wrong. You can see then that when we pair everything correctly, all the colors then line back up together. So, okay. ChIP-seq, I'm going to just skip over this in the sake of time.
Because it works.
It works, and it works at picogram inputs. Yes. So methylation. So Matt did a great job of introducing this. I'm going to pick it up a little more. So methylation, what Matt was talking about, this bisulfite conversion, it's so harsh on the DNA that you can fragment your DNA with it. So when we do single cell experiments, so when you think about doing a single cell, you're putting the single cell in the tube. Now lyse it. How are you going to lyse it? Don't lose the DNA, make the DNA completely accessible. Oh, now you're going to do Illumina sequencing, now you've got a fragment. How are you going to fragment? Well, you can use bisulfite conversion. You'll get a broad distribution, right? You're not going to get that tight peak of inserts. But bisulfite conversion is so damaging you can fragment your entire sample with it. But in that, you then essentially create single-stranded DNA, quasi-heteroduplex. We have a technology that allows us to attach adapters to single-stranded DNA and make a successful library. So what we have is what's called a post-bisulfite treatment library. You have your DNA, you do your bisulfite conversion, and then you come in and you make your library. Traditional method, and I was just looking at some data with Vince, where they used, at least 4 times more DNA than us. One route is you go this way, you make your library, you do your bisulfite conversion, and then you lose 90% of your library. So the way you overcome this is you start with a microgram of input. The method that Vince was showing me, where they also use more DNA to overcome the bias, was random priming. So you do your bisulfite conversion, and then you come in with your random primers, sit them down, and amplify things up. But what you find out is that random primers are biased, actually. But the bisulfite conversion disrupts a lot of the binding of the primers, and so they don't work as they should work. So these Picard plots, here we go again. You can see this is us. This is that traditional route. We have pretty good genomic coverage, very low bias. Remember the frowny face? Right? That's a frowny face. You don't want to see that in your data. It means you're missing a lot of high AT, high GC. Oh wait, you're doing methyl conversion, right? So you're missing a lot of the important parts of the genome that you want to actually detect in this case. And then our complexity, it's something that Matthew was talking about earlier. You can see that our complexity compared to everyone else is actually quite high. We're at a normal 7, and then when we normalize these other guys, they're very low. So how many of those DNA molecules are we actually converting to useful sequencing libraries? CPG giant, this is a way of just enriching for those CPG islands, regions within the genome. This is made by Roche. And here, what we're doing, it's an 80 megabase target, very, very similar to using an exome. But what we're showing here is 100 nanograms for ourselves and 1 microgram for kappa. And the data is pretty much indistinguishable from each other. So we're using 10-fold less input. And if you start thinking about what you're trying to do in bisulfite conversion, you want small cell populations because you want to minimize your diversity. So here's our data at 10 nanograms, and you can see that kappa has essentially gone off the rails. That's what their mean coverage is 1x. Well, we're at 35, and then we can actually get fairly usable data all the way down to 1 nanogram inputs. We show this to people, labs, early access labs. Again, they don't read the manual, and they go straight to picogram inputs. So we're good down to about 100 picograms. What we're finding after 100 picograms is we're having to modify our protocol. So that's definitely work.
Why do you pull down? You pull down the line
For the Roche,
Yeah.
you do the conversion prior to Roche.
Doesn't Roche then also say design every different converted base so the probe?
The probe density is outrageous. Yeah, it's adjuvant that does it the other way around. Yeah, and so I'm going to skip over this slide. This is when I'm going to start going fast. This is work done by Dennis Lowe. Remember I was talking about liquid biopsies? So we're going to take the circulating free DNA, but what we're going to use is we're going to use hypomethylation, the global methylation state of the sample to detect cancer, not an individual point mutation. So it's a 10 million read assay. It's a very light assay, and we only need 5 nanograms of input. And this slide, what we're showing is about how much DNA you get. So on average, you get about 5 mils. So we're getting about 15 nanograms of input from a sample. That's it. 5 nanograms into the conversion.
What are you going to use for conversion?
Zymo. Cambridge won't work with us, so we want to. It's the time difference.
See when we get everybody together?
It's evil. Recall we're doing low coverage, right? So this idea of low coverage, I'm going to show one more example for endangered animals, how low coverage worked. We're only doing 10 million reads, so that means you can't really look at any specific gene and say, do I have hypo or hypermethylation? What's interesting though, in this one case, we can see that P53 is hypermethylated, right? So it's turned off while all the other genes are turned on. But when we look at the entire genome, this is the effect you get. So our normal is in green on the outside, and then our individual with cancer is in red, showing the hypomethylation. 10 million reads each, so on a MySeq, we can put half a dozen, and I think it's 4 samples on the MySeq run. So we can do, so it's a very powerful way of doing it. We did 8 different patients, or samples I should say, from 8 different patients, all with various different types of cancer. You can see in some cases we had very low percent hypomethylation, 0.4%. And in this case, this very sick individual, they had over 40% hypomethylation, so very relevant. So again, you're looking at the global methylation state as a biomarker to see what the cancer burden is within the circulating free DNA. Took those same samples, we ran it across 56G to look at point mutations. We have 4 samples. We had their normal adjacent, we had their primary tumor here, and then we had their circulating free DNA. And you can see that we're getting good concordance in some of the samples. In other samples, you can see that in this individual, the PIK3CA mutation went away, but unfortunately, they picked up a P53 mutation. Not a good situation for that individual. This one, the BRAF, 23% and down to 1%, so hopefully their treatment's working. Only think positive on this.
So circulating free DNA is post-treatment, that's what you mean when you say?
This is actually, these individuals...
It's not just undetected in the circulating free DNA, or it's actually that reflects a remission of some kind?
It's a time point. So it's one time point, so you can't say either way, right? So what you need to do is a longitudinal study. Ideally what you want to do is someone before treatment, right? See what their baseline is, then
right, so
post-treatment.
The 0% PIK3CA versus the FFPE tumor, 17 to 0%. Was that tumor circulating free DNA taken at the same time?
No, this is taken after the resection.
After resection. Okay.
Right.
Okay. Got it.
Good question. I'm sorry, I'm going very fast. 1S technology, I highlighted this for in the methylation. This is the engine behind our methylation. This is how it works. We have our own technology that comes in. We use a truncated adapter, come in and we tail the fragment. It's a single-stranded DNA. We do a polymerase off of it, and that allows us to add the second truncated adapter, and then we put on the full-length adapters in the end. So this is how you deal with single-stranded DNA.
How efficient is that?
It's very efficient, but we have at high AT, high GC, you do pick up some biases. It's a very fast workflow, though. It's a 2-hour workflow. This is doing viruses, metagenomes, looking at some metagenomes. This is expected. This is one case here. So we're being pretty faithful to the metagenome. This is the expected for the second set of controls. Again, we're being very faithful there. And then here the and another expected and what we're pulling out there. All right. How often do you get to boil your DNA in sodium hydroxide? Why would you ever do something like that? Well, if you're working on microbes, you're working with extremophiles, extreme bugs that live near, say, a lava vent on the bottom of the ocean. They're pretty hard to crack. So you can do bead beating, and this takes hours. And you treat your DNA very gently. Or you just boil it in sodium hydroxide and be done in an hour. The DNA is pretty much useless, I'll tell you that, so don't do it unless you're going to use our technology. So when we do that, what you see is, again, what we have the bead beating here, sodium hydroxide here. We're doing a de novo assembly, 42 contigs versus 46. If you do enough de novo, those are pretty much equivalent assemblies. When you run a de novo assembler, if you run it multiple times, you can actually get multiple answers. It's loads of fun. You can see all the subgroups, the functionality within the genome, and those are all equally represented too. So we've got good, faithful representation of the genomes. And shockingly, there's a lot of people now using sodium hydroxide to boil their DNA out of their bugs. Okay, last 3 slides, I promise, and they're my most favorite. So we did some work with NOAA, Southwest Fisheries out of La Jolla. And we're looking at the vaquita. It's a porpoise. It's a solitary animal. It means Spanish for little cow. It's what it looks like. And there are about 100 animals left in the world. And they're all within the Gulf of Mexico here. Oh, excuse me, Gulf of California, shown here. So they're getting killed by gill nets and things like that. So as California, our great state of California, likes to eat their sushi and shrimp and other things, they're harvesting out of the Gulf and they're capturing these guys. So the idea is, well, you do genetics to actually understand the animals, right, to function out, right, what makes them so special, what makes them so different. And also it's a good way of proving to the government and others that they are indeed a different species. So we worked with these guys, but NOAA was very reluctant. The samples are very precious, as you might imagine. You're looking at animals on the verge of extinction. You're not going to get more samples. The way they get these samples is grisly, right? Animal gets caught in a net, ends up on a fishing boat, stays there maybe for a day, gets to the dock for a day. They call someone, a biologist, say, hey, I think I got something special here. The biologist comes, looks at it. It's now 3 days sitting in the sun. Maybe it washed up on the beach after hanging out in the ocean for a week or 2. So the animals are decaying, sitting in the sun. They come in actually with bolt cutters. They chop off a piece of the animal, usually the jaw. Then they drop it into a jar of, you would hope, ethanol, but not necessarily ethanol. And then they put it on the shelf. And it will sit on the shelf for 30, 40 years. In our case, in this case, it's 34 years. 30 to 40 years, in some cases it's up to 50 years. The best part is that solution changes over time, that they get ideas, oh, this is another way. So we're talking about highly degraded DNA. And when we started working with these guys, they had low hopes because they were essentially getting 80% fail rates. They were, and even high, they didn't even try the really degraded samples. So what they gave us is they gave us 12 not endangered porpoises that we then sequenced and showed them that we could work. And then they gave us 12 more where they were really degraded. We're talking 40, 50-year-old specimens. And our 2S technology failed. And so we went to the 1S technology, used that, and they were giving us a lot of DNA. But what was interesting about the DNA, and I didn't really belabor this through mine, is how you quant DNA. Hopefully you've spent hours talking to them about this. In our book, there's only one great way of quanting DNA. That's qPCR, right? Nano, if you use the nano drop, please don't ever send me DNA because it's wrong. The bioanalyzers also, right, that will accept that. The nano drop, not so good. So we were getting lots of good DNA, or high DNA concentrations, but it ended up that depending upon which technique you used, you got a different answer. If you let things incubate for an hour or so, you got a different answer. So it told us that the DNA was it was very degraded. So we didn't have enough. 1S though, we were able to make libraries and we had 0 failures. Now what we did is we took 12 samples, so their genomes are about the same size as a human genome. We could take 12 samples and we could run it on the mighty MiSeq. From that, you can't do anything on the nuclear DNA, but what you can do is you can reconstitute the mitochondria. So on average, we were able, right, because your mitochondria genome, it's a lot smaller, but it's in higher copies relative to the nuclear. So when you do that low pass whole genome sequencing, you're almost always getting the mitochondria, and you're getting it well. And so in all our cases, I think the lowest case, it was about a 20x coverage. On average, we were about a 40x coverage of the mitochondria. And that then allowed our friends at NOAA to put together this tree. We didn't do any of the beluga or narwhals. We did many of the porpoises that are shown here. And you can see here's Vaquita, and indeed it has its own branch representing its own species. And that is all I have. So I'm done. Unless you want to see the other 60 slides. We're done. It's beer drinking. There's still beer out there, still in the fridge. No, you drank it all?
They only gave us like a 12 pack.
Wow, they're cheap.
Yeah, for 25 people, we're sharing.
Have you guys gotten, or, Neanderthal, I mean, the other big,
Ancient DNA?
ancient DNA?
So we have done some ancient DNA work. The problem with ancient DNA, especially when you get into Neanderthal or things that are 20, 30,000 years of age, is your DNA fragments get so short that now they're too short for our SPRI cleanups. And so that's the issue we have. So you're looking at inserts about 30 bases in length. And so that's somewhat problematic for our technology.
Why not just clean up the library as opposed to the DNA? This is what same thing for microRNAs. Like, so.
That's what I'm talking about. That, right, as you do the library prep, there's a SPRI in it, and what we can't do is distinguish between the adapters and.
But if you can't do SPRI, then you got to do so. I can show you some methods that we're doing to distinguish the smaller fragment with the smaller adapting dimension versus an insert.
Perfect. We just don't do too much ancient DNA.
Well, if you got some US library, send that away and we'll work with
All right. Now I've got a name to send to you. All right, I've kept you up way too late. Thank you for being so patient. Enjoy your time here and torture Vince as much as possible. Be nice to Sean, though.